Okay, that happened sooner than I thought. Let's continue on now. So one of the things that we need to understand to look at the distribution of traits and alleles and genes and chromosomes is a little bit of anatomy of a chromosome. So each one of these lines would be a linear chromosome from a eukaryotic cell. One, let's say, would be from mom, the red one, and the other would be from dad, that would be the blue. Whenever we see each individual stripe here, that's the location or the locus or loci for multiple here of given genes. So this would be gene A, gene B, and gene C. Each gene in this particular case is coming in two different flavors or alleles. So over here you have big A, so you have two copies of the same allele for gene A. Here you have big B, little b, so now you have opposite copies or different copies for the same gene. And here you have little c, same copies for gene C. When you have two capital A's, we call that homozygous for the dominant allele or homozygous dominant. Two copies of the little c here would be homozygous for the recessive allele of that given gene. Here, big B, little b, this is heterozygous for the B gene. So when we say homozygous, you want to say homozygous dominant or recessive. Is it the dominant gene or the recessive gene? Because that's going to make a difference here pretty soon. Heterozygous means that you have one copy of each. So hetero means opposite and homo means same. So what you're looking at here is just basic anatomy of two chromosomes. And right here, these little skinny parts are the centromeres. That's going to become important when we talk about mitosis and meiosis. Sex chromosomes, X and Y, are not homologous. X is really long, Y is really small, but the tips actually are homologous. They're called pseudo-homologous regions, and that'll become important again in a little bit. And those are the short regions of homology that you'll read about in the book. Now, cellular division. Cellular division happens in different ways depending on if you're a eukaryote or a prokaryotic cell. Basically, the purpose of any cell division is for asexual reproduction. In other words, the cell is just copying itself. It's cloning itself. And so, for some simple organisms like bacteria or archaea, or even amoeba and yeast, you'll see that they undergo a certain type of division that can be more complex or less. What am I talking about? What we're talking about is basic mitosis. For a cell to become triggered to divide, usually the environment is going to be sending a signal saying, hey, you need to get ready to duplicate all your DNA. Cell division, in addition to allowing a cell to copy itself and clone itself, so let's say one bacteria divides into two to make two bacteria, this is also important for increasing cell number in an organism. So one of the interesting similarities in all living organisms is that our cells, for the most part, are roughly the same size. So all the cells in a human and all the cells in a whale are about the same size. It's just whales have more of them. And so additional rounds of cell division increase the number of cells in a multicellular organism, plants and fungi as well. So humans start out, let's say for example, and all animals start out as a single fertilized egg, and the adult stage is the consequence of trillions of cell divisions that occur. Now, let's back up a little bit and start with the simplest type of cell division, which is called binary fission. Binary fission is what allows bacteria to divide very, very quickly. So, for example, E. coli can double its population every 20 minutes because every 20 minutes a single cell can divide into two cells. Before that, though, all the chromosomes need to be replicated. So in the case of E. coli has one big round chromosome. That chromosome has to be copied so that each new cell gets a new copy of that chromosome. That particular type of division is a little bit simpler than what we're going to be looking at in eukaryotic cells. So let's look at this. The mother cell is the original cell. Here's the bacterial chromosome. The bacterial chromosome is going to become copied, and the cell will start elongating. As it's elongating, the two new copies start separating apart, and a new protein is produced, the FTSZ protein is produced, to allow a division or septum to develop between the two opposite sides of the cell. At this point, each side of the cell has a copy of the chromosome, and the septum continues to pinch apart until you get a full formation of a full septum. After that, two cells exist, and these two cells are called daughter cells. I don't know why they're not son cells. I haven't looked into that. I have been asked that many times. I have to remember to go look that up. But they're called daughter cells at the end of the division. So binary fission does not involve any fertilization. In other words, binary fission is only allowing the cell to copy itself and make an as genetically identical individual to itself as possible. Yes, there might be some gene mutation that occurs, but for the most part, you have two genetically identical cells. A more complicated type of cell division is called mitosis. Mitosis is the type of cell division that gives rise to many, many new cells and is more complicated than binary fission. And one of the reasons is because in eukaryotic cells, we have that nucleus that houses DNA. And furthermore, we have additional DNA chromosomes. So remember, bacteria only have one chromosome. In many eukaryotic cells, you have, let's remember in flies, you have 8 chromosomes. In dogs, you have 76. So eukaryotic cells are destined to undergo through a whole process of steps to organize chromosomes and prepare the cell for division. And this process is called the cell cycle. The cell cycle is actually broken down into 4 phases, although you may be very familiar with one phase alone. Let's start with naming the first 4 phases. G1, which is gap 1. During G1, cells are kind of sitting there doing their job, all right? They're not necessarily getting ready to divide, but they're performing metabolism. They're doing what they're supposed to be doing. At this restriction point, if the cell is given a signal to divide, the cell is going to commit to division. We call this actually the G1 checkpoint. After that, the cell is committed to go into S phase, or DNA synthesis phase. During the DNA synthesis phase, all chromosomes are copied. So you'll see in this particular case, you have 3 red chromosomes and 3 blue. They're going to copy so that in the end, you have 6 red and 6 blue. And what's interesting is, let's say you have a single chromosome in G1. By the end of S, that chromosome is going to be doubled and attached to its partner or its copy right next to it. We call those sister chromatids. After gap 2, or during gap 2, I should say first, all the new DNA that was synthesized is checked for spelling errors, okay? Checked for mutation. And if mutations are found, those mutations are fixed. Our cells are really good at fixing mutations. The fact that we even get any mutations is actually kind of crazy, but we'll talk about that another time. At the end of gap 2, which we don't have drawn in here, is another spot, another pause that the cells go through called the G2 checkpoint, where that spell checking is happening, and the cell is growing a little bit. It's putting on a little bit more cytoplasm in preparation for what we call M phase, M for mitosis phase. Mitosis literally means the division of the nucleus, all right? A cell can undergo mitosis without undergoing what we call cytokinesis, or pinching apart. So what you're probably familiar with as what most people call the cell cycle or cell division, which is prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. This that you're most familiar with, looking at little X-shaped figures and stuff, is only M phase. The cell cycle is G1, S, G2, and M together. Why is that? Well, it turns out that during the 19th century, 18th century even to some degree, microscopes were good enough that you could actually see the nuclei of cells, but you couldn't see what was happening very well. And so this whole region here, G1, S, and G2, were originally called interphase. So you can be very general and just call this interphase. With the advent of nuclear technology and fluorescent stains and better microscopes in the 20th century, we were able to designate gap 1, S phase, gap 2, separate from mitosis, and we actually know how long each one of these phases lasts during the life of a cell. So remember, interphase equals G1 plus S plus G2. M phase, mitosis phase, is something all in and of itself. That is prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So what are we looking at here? Cells that are actively dividing are going to be going through G1, S, and G2, and you won't even notice it. But once the cell hits that M phase, the mitosis phase, all of a sudden, you'll see the chromatid pairs. Now, there's one other spot that I didn't really mention very much. Let me go back to this former picture. You can also find a little spot sticking out the side here called G0 or G0 phase. G0 phase is a spot where a cell will sit and will never divide again. Basically, we call these terminally differentiated cells. In other words, they're not going to change. So neurons in the brain, neurons in the spinal cord, not necessarily neurons in the peripheral nervous system. These cells don't undergo cell division after they've been born and after they've terminally differentiated. And there are good reasons for that we could talk about for a day. Maybe we'll have a little office hour period that we can talk about that. But moving on in mitosis G1 phase, the cell receives information about, are you going to go divide again, or are you going to do what you need to do? If the cell commits to dividing, it's going to jump into S phase, where the chromosomes are replicated or duplicated. Later on in the semester, we're going to talk about how this happens. But basically, a single linear chromosome gets copied, and it sits right next to its sister over here. They are joined at what we call the centromere, and these two yellow points are called the kinetochore. The kinetochores are extra proteins that hold those little belts together, the centromeres. So, some people do call these individual chromatids, but only when they're in the X shape holding onto each other. Once they're separated, they're chromosomes again. So these are sister chromatids. So when we're looking at a human karyotype, really what you're looking at are chromatid pairs. So this is what they literally look like. So this is one chromosome, and this is its chromatid partner. And during mitosis, they're separated from each other. They go to opposite sides of the cell, and they become a part of the new nucleus. And so at the end of S phase, each cell has twice the number of chromatids. So you started with 46, all right, 46 sticks, and then it doubles. During late G2, spell checking happens, and then the cell goes into mitosis phase where the two are separated. And so let's look at what that looks like. I'm going to scroll through some of these and rely on you to read through those slides. So during interphase, you kind of can't see what's happening in the spaghetti, but the one thing you can see is the duplication of these two little structures called the centrosomes. The centrosomes are going to send out microtubules to contact the kinetochores of the sister chromatids and pull them apart. It's kind of like, almost like, sending out a fishing rod, catching a fish and pulling it back to the boat. During prophase, these microtubule forming centrosomes are migrating to opposite sides of the cell, and the nuclear envelope starts breaking down. You can visualize the pairs of sister chromatids. So these are the events occurring during prophase. During prometaphase, which we're going to talk about more when we get to meiosis, because it's a little more important there. The chromatid pairs are getting pushed to the center so that they end up lining up one on top of the other. So in eukaryotic cells, in human cells, you have 46 sister chromatid pairs lining up right through the middle, okay? By the time that they get there, there's prometaphase. We call that metaphase, all right? At this point, the nuclear envelope is gone, totally dissolved. Now, anaphase is where those microtubules from the centrosomes start pulling the chromatid pairs apart. Like I said, it's like fishing, putting out a fishing line and pulling back the fish or the chromatid. During telophase, new nuclear envelopes are forming around opposite poles, and the formation of the cleavage furrow is initiated. So in other words, the cells are starting to pinch apart, and two new nuclei are forming. And then each one of these centrosome pairs are degenerating back down into something that can become prepared for the next cell division. And so in most cases, mitosis is very, very quickly followed by cytokinesis, or pinching apart. In animal cells, that involves the formation of the cleavage furrow, and in plant cells, it's the formation of the cell plate. So here's an example of the cleavage furrow in an animal cell. And I unfortunately don't have a picture of the cell plate. I'm going to stop here before we start up with meiosis.